let's start semi-conservative model. Okay. So the scientists Michelson and Starr, okay, they use E. coli as the model organism. Okay. So what is the model organism? E. coli is the bacteria, right? So they use E. coli to prove that the DNA replication is formed because of the semi-conservative mode. Okay, I am already told you that the replication, DNA replication is done by semi-conservative model. So, see, first there are three modes of replication, okay, which was introduced during 1950, like conservative, semi-conservative and dispersive. Okay, so what happens in conservative? See, this is the parent DNA, okay, and after replication, what happens? The parent DNA replication means what? Means there is a division of cell, correct? In cell division, what happens? One cell is get divided. Okay, means what? The genetic material is also divided, right? What is the genetic material inside it? DNA. Okay, this is the DNA. In conservative model, what happens? The parent DNA means the DNA which is in the parent cell is transferred into in one cell, okay, and the copy of that cell, copy of that DNA is transferred into the next cell. This is nothing but the conservative model, okay, means the whole DNA, whole parent DNA is transferred into the one cell and the daughter DNA is transferred into the next cell. This is the conservative model. So the next model is the semi-conservative model. What happens in the semi-conservative model? This is the parent DNA, right? After DNA replication, what happens? The strands are get separated, right? And the strands are get separated, and the new strands, the new complementary strands are formed on each strand. Okay. Means what? 50% genetic material is transferred to the first cell and 50% genetic material is transferred to the second cell. Means this is the semi-conservative, right? Yes. Got it? Now the dispersive. Dispersive means what? This is the DNA. After replication, what happens? It breaks into part. Okay? And after breaking, it joins to the new part. Means what? Suppose this is DNA. The strands are separated. Okay? After replication. And after replication, what happens? These strands are cutted. Okay, they are break and there is a formation of new patches or new strands. This is a dispersive method. Got it? Means the whole DNA is not transferred from parent to child. Means they are transferred in partitions. Okay, the genes which are located on the DNA are get dispersed. Okay, this is the three modes of replication: conservative, semi-conservative and dispersive. So the researchers were confused between these three, that what could be the actual pattern of DNA replication. Okay, because there are three modes of replication. Which one is the correct one? So in 1958, the Michelson and Franklin Stan, okay, they presented their research where they concluded that the replication of DNA is a
Semi-conservative
type. Let's see the experimental. Experimental confirmation of semi-conservative DNA. Okay. So this is the method. Okay. And because of this, they prove that the DNA, the mode of replication is due to semi-conservative. Okay. So it is first given by Matthew, Messelson and Franklin in 1958. Okay. They use light and heavy isotopes of nitrogen. I have already told you that there are two isotopes. Okay. Radioisotope. Okay. And heavy isotopes. Right. Radioisotopes, which is where we see it. Radioisotopes.
X-ray rays.
In which experiment? The last experiment, right?
Yes.
Hershey and
Chase.
Okay.
Yes.
Where we use what? 32 phosphorus and 35 sulfur. Okay. Here we are using heavy isotopes. Okay. Heavy isotope of nitrogen and light isotope of nitrogen. Light means normal. Okay. Means normal type of nitrogen. See. Which experimental material used for this experimentation? Okay. First they use, as I already told you, they use E. coli bacteria as a model organism. Okay. Then they use light and heavy isotope. Light means the normal atomic weight of nitrogen is 14. Okay. That is normal nitrogen. Okay. And the heavy isotope, that is the atomic weight is 15. And the third is NH4Cl, means the medium on which they grow the E. coli. Okay. Next, the equilibrium density gradient centrifugation. Due to this technique, they prove that the DNA replication is done by semi-conservative model, and they use the medium, that is cesium chloride. Okay, which is density gradient medium. Okay, which is a density gradient medium, which density is equal to the heavy isotope. Okay, which density is equal to
Heavy isotope.
Heavy isotope. What happens? See, they first take E. coli. Okay. They first take E. coli in a petri dish. Okay, what they do first? They take E. coli in a petri dish containing what? NH4Cl medium for their nutrition. What they take? They first take E. coli on the petri dish, and inside the petri dish there is a NH4Cl medium, that is ammonium chloride medium. Okay, and they allow them for several replications. Okay, they allow the E. coli for several generations, and after that E. coli, okay, and run on the centrifugation. That is the density gradient centrifugation. Okay, that is the density gradient.
Centrifugation.
Understood?
Yes.
What they first take? They take E. coli, okay, and put it on the NH4Cl medium. After several generations, the E. coli bacteria are grow, right?
Yes.
And after that, they take out DNA from the E. coli and run on the density gradient centrifugation. समझ में आ रहा है?
Yes.
Okay, means what? DNA separate किया E. coli से, okay, and added into the density gradient medium. That is CH-Cl2. ये टेस्ट ट्यूब जो है इसके अंदर क्या है? CH-Cl2. ओके, ये मीडियम है. कशासाठी? फॉर व्हाट? टू डिटेक्ट डेन्सिटी.
Density.
Okay, this test tube contains CCl2, right?
Yes.
This is nothing but the density gradient medium. Okay, ये DNA इसके अंदर ऐड किया. Okay, after adding the DNA into this medium, what happens? They run it on this technique. That is centrifugation technique. We have already seen that. Centrifugation technique means the device, okay, contains the test tube holders and they run with the centrifugal force. समझ में आ रहा है?
Yes.
Okay, centrifugal force in the Harshand Chase
Chase
experiment.
experiment.
Okay, same. To note down the density gradient medium, they put the DNA inside the test tube.
Test tube.
And what happens? This is the high density. Means the density, note down, it should be high. Why? Because of the 15N heavy isotope. We provide it NH4Cl, okay, but the N inside the medium is containing heavy isotope, that is 15N. Clear?
Yes.
Means what? The E. coli is grow on the medium containing 15N heavy isotope. Means inside the NH4Cl we are providing them 15N, that is heavy isotope, right? And then the E. coli grow on that heavy isotope. Okay, the DNA is taken out from that medium, from the petri dish, and added into the test tube.
Yes.
Containing the density gradient medium, that is CaCl2. After centrifugation, what happens? A band is occur. Okay, a band of high density is occur at the bottom of the test tube. The band of high density is nothing but the band of 15N heavy isotope. This much clear?
Yes.
Got it? Okay. Now, what happens? This is the first point. Okay. Here we see the DNA from the E. coli, okay, having 15N isotope. 15N heavy isotope in their medium, right?
Yes.
This DNA occurs here at the bottom of the test tube. Okay, the band is occur at the test tube, that is 15N band, that is of high density. Clear? Now they allow that E. coli in the next generation. What happens? They allow the E. coli for 20 minutes in the medium containing 14N isotope, that is light isotope.
Okay.
Okay, after 20 minutes means in the generation first, they allow the E. coli to grow in this medium, means 14N NH4Cl, means the N containing 14, that is light
Light
isotope.
isotope.
Okay, then what happens? After 20 minutes, the DNA replicates. Okay, the DNA gets replicates. After replication, what happens? Now the E. coli is on the medium containing 14N isotope, and before they are in the 15N isotope. Okay, in the next generation, what happens? The one strand, okay,
From 15N.
is of
15N.
15N, and one strand is of
14N.
14N. Means what? They take out that DNA, same in the petri dish. They take out the DNA from this E. coli, which is on the light isotope ammonium chloride.